जी
That's why मैं
मैं सर आपको ये कर दूंगा ना, डॉक्यूमेंट कर दूंगा व्हाट्सएप पे।
हो जाएंगी। ये थोड़ी लेंथी तो नहीं होगी, इतनी ज्यादा नहीं होगी वैसे।
सर जी लेंथी होती है लेकिन डॉक्यूमेंट हो जाएगी।
अच्छा,
हाँ जी।
बताना अगर कुछ। आई डोंट नो अबाउट दिस हाउ it is possible. Normally I think in long videos, specifically individually, I think they are not we're not able to send, but it's okay if you know about it.
नहीं
मुझे
सर,
फिर
आप
आप ना इसको
एज अ डॉक्यूमेंट हो जाती है, मैं आपको कर दूंगा।
एज अ डॉक्यूमेंट इन व्हाट सेंस, लाइक मैं उनको एक्सट्रैक्ट कैसे करूंगा?
सर जी वो आप वो डॉक्यूमेंट को टैप करेंगे तो अंदर वीडियो होगी, वो आपकी गैलरी में सेव होने का ऑप्शन आ जाता है, वो आप सेव भी कर सकते हैं।
ओके फाइन, fine, that's good. So let's start from the structure of virus.
ओके सर।
So in this case, I have told you that this virus is actually a non-living organism, but once it gets into the cell of any living organism, like bacteria, like other living organisms, like human,
यस सर।
so they take over the machinery of the uh, living organism or that specific host.
ये
That's why we call it that it is a living,
यस।
because inside it was just a crystal. It was not reproducing, it was not dividing. But once it gets into the cell of an organism, so there it started division. How? Because it will take over the machinery or the protein synthesis machinery of the bacteria or any other cell, and it will start to produce its own structure. It will start to synthesize its own structure.
ओके।
So that's why I call it. So the size of the viruses which are mostly considered, that is about from 20 nanometer to 300 nanometer.
ओके सर।
300 nanometer. So this is the range of, uh, you know, range of uh, the size of the viruses.
ओके सर।
And I have told you that virus is not a cell. It is not a cell. I use the term here acellular organism. Acellular. Acellular means not having any cell.
ओके सर।
Cellular. This is acellular, right? So you can also write here that the cell is not present in case of virus.
ओके सर।
So it do not have the cellular structure.
ओके।
Similarly, we can also say do not have metabolism. There is no metabolism in this case.
ओके सर।
It has not the ability to synthesize the substances by itself. It has not the ability to break down larger molecules into smaller ones. So that's why there is no metabolism. All viruses are parasites. Remember, all
वायर
viruses are
पैरासाइट।
parasites. And you know what are parasites?
नो सर।
Okay. Parasites are actually, it is, they have the association with their host. There is a host, and in there must be a specific host with which these parasites or the viruses have some association. What type of association? These viruses, these viruses in the form of parasites, they get shelter from the host,
ओके सर।
and these viruses get food from the host, right? And in return,
यस
and
सर।
in return, what they do with this host to infect it or to cause disease. So any organism that harm the host
यस सर।
while living in that organism, and get from there. Actually, you can say parasites are just the guests and they have to live on the some specific host. And on host, actually, they are getting shelter and food, and in return, what they do with that host to harm them or to cause disease.
Yes, sir.
All right. So this is a function of parasites. Now, parasites, we commonly use the word pathogen. There is another term that we commonly use, pathogen. Pathogen. Those organisms which have the ability to cause disease,
Okay, sir.
right?
Yes, sir.
So this parasitic association, this parasitic relationship is actually in which the parasite gets shelter and food from the host and in return to harm it. That is, we call it the parasite. Now, now we can talk about its structure, its composition. Uh, there are two important things in it: protein coat,
Yes, sir.
protein coat, and the second important component, that nucleic acid.
ओके सर जी।
Nucleic acid.
सर जी वो
Uh,
आपने वाइट बोर्ड का नहीं देखा कुछ?
nucleic acid. Are you getting this point?
यस सर।
जी
वो आपने वाइट बोर्ड नहीं देखा उस पे
जी।
वो बोर्ड पे वो
Uh,
आपने
जो
कहा।
जो जूम के ऊपर है, लास्ट टाइम मैंने उसको ट्राई किया था आपके साथ, बट मैं इसको ना आई विल ट्राई टू टेक सम डायरेक्ट फाइल। असल में ये स्क्रीन के ऊपर मैं तो चूंकि ये इसलिए यूज करता हूँ, ये स्लाइड्स हैं, सो मुझे ये थोड़ा सा बेहतर ऑप्शन लगता है, दैट्स व्हाई कॉमनली यूज है।
सर जी
अदरवाइज ये है। जी
थोड़ा
जी।
कंफ्यूजिंग सा हो जाता है वो, जब मैं रीकैप लेता हूँ ना तो बड़ा कंफ्यूजिंग सा सारा हो जाता है, इसलिए मैं कह रहा था उस पर ज़रा एक प्रॉपर पैटर्न में ऊपर से नीचे आ रहा होता है।
ओके ओके। वो एक्चुअली ना आई हैव टू मैनेज के वो देखना पड़ेगा कि जो नोटबुक है पीछे एक ओपन होती है, जो कि मैं कॉमनली वो यूज करता रहा हूँ, जो सर शेरोज के पास है, वो मेरे पास ही था पहले। उसमें यही है कि उसमें ये होता है कि हम थोड़ा नीचे पेज को थोड़ा लेंथी कर सकते हैं।
यस
यू
सर।
राइट इन दैट केस एंड आई विल डू समथिंग लाइक दैट कि मैं उसको ना कोई उस तरह से करना। अदरवाइज ये वर्ड फाइल में भी ऐसे हो जाना चाहिए, कोई इतना ये वो नहीं है, बट आई विल ट्राई। मैं इस वीकेंड पे होपफुली इसको मैनेज करूँगा।
ओके ओके सर।
राइट फाइन। सो
ये
ये ठीक है कि आपने रिमाइंड किया।
यस।
एनीवे, सो नाउ we can talk about another thing. I have told you that there are two things, like one is protein coat. How? Like, for example, you have a cell like this. So, in this case, virus is commonly, there are many types of, for example, one thing I have told you that there is a protein which is present here. Other
यस
thing, mostly they may have DNA or RNA,
यस
right? So the any one of them, not both, one of them, nucleic acid may be present in the viral uh, uh, structure.
यस
So, for example, there is a coat, because we have talked about there's a protein code. So this is the protein code. Most of the time, there are few structures on it. Some glycoproteins attachment. You can say they are attached uh, on this protein structure. On this, we can say a protein code that is called capsid. This is actually a capsid. We call it, this protein code is called capsid. So this capsid may have some protein structures like this on its surface,
यस सर।
like this, in a whole body, like this. So the whole structure is, you can say, these are somehow, for example, on this place, there may be a glycoproteins.
Okay,
The
sir.
composition of these spines, actually, these are just like a spines which are spreading outside of this body. So it is actually composed of glycoproteins.
Okay, sir.
Got it?
Yes.
So another thing about which we can say some, for example, definitely this membrane is a lipid membrane. Uh, for example, this is a protein structure which outside, and they may also have some lipids inside.
Yes.
So with this all, uh, for example, if there is a RNA, ribonucleic acid virus,
Yes, sir.
there will be some RNA here. So for this, somehow there is another uh, enzyme is present that is called reverse
Transcriptase.
transcriptase, right? So
Yes.
reverse transcriptase is also present here. So for this is what, this is a structure which is present here. It is not double. Remember, it is a single. Uh, uh, I should fill this. Okay, so it is not double layer. It is just one structure. So this is an issue here. Yeah, this space storage issue. So anyway, so here it is. I can label it here like it is R and A. Right?
Yes, sir.
So single protein in capsid is called what? Capsomer. Remember.
Okay, sir.
Cap. So this capsomer is actually, it is a single protein structure which is present in that is forming this whole complete capsid. This capsid is actually formed by the subunits. We call it, it's a subunit, and this subunit, this subunit is actually combined with one another, and this capsid is a, this capsomer is a single protein. So number of proteins they combine with each other to form a capsid, and that capsid is actually the outermost layer or the protein structure of the virus. So this was the short introduction of the virus. We are not going to in detail, and it's not our requirement here. We just need to have an idea that what virus is and how its structure is made up of. You can say there are two main components of the structure of the virus. One is the nucleic acid that may be in the form of DNA or RNA, and the second thing is protein code, and that protein code is actually containing nucleic acid inside of it. Okay,
Okay, sir.
so that protein layer is called, what it is called, it
Aspect.
is called capsid, and
Yep.
this capsid is made up of smaller subunits, and those subunits are called capso. Okay,
Okay, sir.
are you clear?
Yes, sir.
Fine. Now another important thing, cell structure is involved in the protein synthesis, as we have discussed many places and many topics, that the whole this process is the structures which are present inside, they are actually assisting to one another, you know, to synthesize the protein, right? So this protein synthesis is the major process which is taking place inside of the cell. So we just need to have an idea that how is synthesized, or these subcellular structures which are present inside of the cell, how they are involved in the protein synthesis. Are you getting? So
Okay.
just one more point that we need to discuss, and that is uh, the method how this protein is formed. Okay, So
Okay.
protein is synthesized. We call this topic, we can name this topic cell structure involved in protein synthesis.
Okay.
Cell structures
And
involved in
protein synthesis.
protein synthesis. Cell structures involved in protein synthesis. It means we will just have a glimpse on those small structures, subcellular structures, which are involved in this uh, polypeptide chain that started from where we know very well there will be the final product that will form and which will further modified, but that is a polypeptide chain. We just need to know how this polypeptide chain is formed. So here you will just recall your previous knowledge that you have up till now, and you have studied about that in whole this in entire of this chapter. So what was that? So from where you will start, or is what do you think? Protein synthesis, the first step in protein synthesis was what do you think? For example, you have a cell here, right? So there is a nucleus here, and we have discussed in detail about the structure of nucleus, how it is involved in protein synthesis, and how other structures which are present inside of the cell, how they are involved. For example, we are talking about this nucleus.
Yes, sir.
As we know, there are two things here. There was the nucleolus inside,
Yes.
and we know very well this nucleolus was the site, was the point where ribosomes are formed, and these ribosomes, once they synthesized here, then they will move from this nucleus to the outside into the cytoplasm. And another thing that is present in this nucleus, that is what? That is DNA, chromosomes
Yes, sir.
in the this DNA. And this DNA, once it will uh, transform into the messenger RNA, this messenger RNA will move from this place to the outside of this, the cytoplasm. So these ribosomes which were synthesized inside, they are already outside here in the cytoplasm. So this DNA, which is transcribed, into messenger RNA. So this messenger RNA is now present outside on the ribosomes. What is its function? You know very well. So this messenger RNA will be read by, yes, will be read by this these ribosomes which are
Ribosome.
present out, right? So
Yes.
once they will read the whole structure of messenger RNA. Messenger RNA is actually the message, and that message is coming from the nucleus, from the DNA. DNA has actually transcribed. We know very well when DNA uh, produces or form the messenger RNA, that is called transcription. We will discuss this topic in detail, but it is called transcription. So once it is transcribed here, right? So then this ribosome will read the message of this messenger RNA and
Yes, sir.
convert that into a polypeptide chain, right? So
Okay.
that polypeptide chain, as we know, there is a endoplasmic reticulum was outside just
Yes, sir.
after the, we know very well. No? So
Yes.
here there are ribosomes are present on this rough endoplasmic reticulum. So there, this polypeptide will further move to the next point. That was what? Okay, what was that? Which structure was here? You
Sir.
can make it more appropriate and proper diagram, but I'm just giving you an idea about this. Like this was the smooth endoplasmic reticulum. This smooth endoplasmic reticulum is outside just after this rough endoplasmic reticulum. From
Yes.
this polypeptide chain will come here, and it will get from one side of this cristae into this structure. And sorry, it is in this case here it will modify into the form of vesicles. These vesicles will move further and go
Yes.
where? Yes,
towards the membrane.
Once these vesicles are formed from the rough endoplasmic reticulums, which are called rough endoplasmic vesicles,
Yeah.
rough endoplasmic reticulum vesicles, where they will go from here. Yes, they will go for Golgi apparatus. You know, Golgi apparatus is also here.
Yes, sir.
Recall that knowledge. So that this is a Golgi apparatus sort of structure, a flattened structures which are present just after this endoplasmic reticulum,
Ye-
and there these vesicles will get into fused with this Golgi apparatus,
Yes, sir.
right? So here there will be the modification. What it will do, there are two functions of this Golgi apparatus. The one function is its function is packaging.
Yes, sir.
It means it will packing. Its main function is to pack that protein which is coming from the endoplasmic reticulum. Second thing is modification. The second point that you can add here, modification, and we
Modification.
have discussed this. Yes, we have discussed this previously, that these will be modified here in the Golgi operators,
Yes,
and from where it moves to membrane in the form of Golgi vesicles. Now, the final product which will come out from these Golgi operators, that will be Golgi vesicles.
yes, sir.
So first, the vesicles which are coming from these rough endoplasmic reticulum, they are called rough endoplasmic reticulum vesicles, and these vesicles will get fused with this Golgi operators. There it will be modified, it
Right.
will be packed and packed into another vesicles, and those vesicles are called Golgi operators, Golgi vesicles.
That is
Got it, man?
process.
So what will happen after this? After this, there will be exocytosis. Are you
Ye-
getting?
Yes.
Exocytosis.
Yes.
What is exocytosis? In exocytosis, this Golgi vesicle will come to fuse with this outer membrane, which is a double layer, double membrane structure of the cell. Here it will invaginate from inside and invaginate to form a ball or balloon shaped structure, invagination. And in this invagination, this vesicle will fuse, and whatever the material is present here in this vesicle, that will go out from this membrane in the form of uh, a transportable structure, whatever it was. So that will move
Yes.
outside, and it will go to another. If this material has to get into another cell, again the whole process will repeat. Like there, all these there will be a vesicle. Those vesicles will actually form from the membrane structure, and then it will go again in the processing. So
Yes, sir.
how here we have just talked about how different structures of the cells, they are involved in the process of protein synthesis. So we
Yes.
have discussed in a very detail that uh, there is a nucleus, and that nucleus actually transcribe the messenger RNA, and the ribosomes which are formed or which are synthesized in the nucleus, they both come out into the cytoplasm, and there the ribosomes are attached to the
Yes, sir.
reticulum and the message from the messenger RNA and convert it into the protein polypeptide chain, and that polypeptide chain will be packed in a vesicles. Those vesicles are called rough endoplasmic reticulum vesicles, and those vesicles will
Yes, sir.
again go to another structure, that is Golgi operators. The Golgi operators will modify it to further pack it, and then it will go to the outside of the membrane, and this is how this whole transportation process will carried out. Are you getting this point? Fine.
Yes, sir.
Just tell me 2 to 3 differences of prokaryotes, eukaryotes, because here this is the main topic. We have to discuss it. We have discussed many times, but I just want you to recall 3 to 4 points so that you must know that there is a very marked difference between prokaryotes and eukaryotes, because we will not discuss these both topics again in upcoming chapters. But here you have to clear your mind. There are two main types of cells. One is prokaryotic cell, another one is eukaryotic cell. How they are different from each other, we how we differentiate them. So for that purpose, you should know at least 3 to 4 points. So the first point, tell me, what is the difference between prokaryotes and eukaryotes? Paresh, what do you think?
Sir, difference in size of
First,
ribosomes.
difference in the, what would be like? P is I'm writing P on one side, U on other side. So tell me, ribosome size, what it is here, 70s or
70
80s?
years.
70. Eukaryotes,
80.
80s.
Yes, sir.
Okay. Other difference.
Sir, histones absent. Present. नहीं, absent.
We can say, no. In case of prokaryotes, naked DNA. There is no histone. There is no wrapped or proteins or present. We can say actually there is no nucleus here. Naked DNA
Yeah.
or naked DNA is present.
And sir,
Okay.
membrane bound.
Hmm. Another point. Tell me another difference.
Sir ji,
Size of the cell. Size of the cell. What was the major range? I think 0.5 to 0.5 to 5 micrometer.
yes,
0.5 to 5 micrometer. It is the range of this. Here on this side, you can say there is not naked DNA, but in this case, nucleus is present. DNA will be present inside of the nucleus. While in case of bacteria, normally DNA is spread outside in the cytoplasm. That is not enclosed in the nuclear membrane, but it is enclosed in the nuclear membrane. This is the main difference between the prokaryotes and eukaryotes. All right. What would
yes, sir.
be cell wall sometimes present, while in prokaryotes, you know very well, in prokaryote cell wall is present. Ribosome
Yes, sir.
is about, yes, size is also different in both cases.
Sir,
We can just
सर जी
use
एक
that.
मेम्ब्रेन बाउंड है, एक
Uh,
नॉन मेम्ब्रेन बाउंड है.
uh, yes, one is membrane bound, other is not membrane bound. Having circular DNA, other having circular DNA. Cell wall made up of murin. The cell wall of the eukaryotes is made up of cellulose. Right?
Yes,
Do
sir.
not have nuclear membrane. Have nuclear membrane. So there are many differences.
Yes, sir.
End of this. I will WhatsApp you a worksheet. I will try to find out here
Yes, sir.
in my. So आपने उसको जरा बैठ के करना है और फिर मेरे साथ काम.
Yes, sir. Okay.
First 5 to 6 minutes में discuss the topic after. I think we have completed our task of today, and we have completed one objective from cell structure, right? So
Yes, sir.
just after this, in coming, we will go on the biological molecules and some of concepts. So this was the whole concept, right? और मुझे आपने वो भेज देना है, वीडियोस ट्राई करें मुझे भेजना, सो दैट मैं देखूं जरा, क्योंकि वीकेंड के बाद मेरे पास भी टाइम नहीं होगा. आई कैन मैनेज कि मैं देख लूंगा उनमें क्या, और फिर आपको मैं वर्कशीट भी भेज देता हूँ. ऑलराइट.
Yes, sir.
Okay, Rez. Thank you very much. Or hopefully we will see you. We'll meet on Monday. Fine.
Allah
That's
Hafiz.
all.
Yes, Allah Hafiz.
Okay.